This is an overview of the functions and features of the AzureSpot Q image analysis software. From File, you can open and close files as well as save files or save files as a file name. When you have multiple images open, you can use Close All to close all images. From Print, you can print the image as it appears on your screen, including any annotations and analysis, or print image info to print the image alongside its image information. Tools allows you to create an animated file of your image or a Z-stack of your image. Create Multiplex allows you to take multiple files and layer them to create a multiplex image. Create Single Channel allows you to take a multiplex image and separate it out to different images for each channel. Generate Report will generate a report of all your analysis performed on your image, including annotations. Clicking the Help button will bring up the AzureSpot Q user manual. Settings allows you to access the general and analysis settings of the software. And this key allows you to access the license information for your software. When you have images open, you'll see this left-hand bar that includes all images that you currently have open. You can close images from this bar or hide the bar in order to see your analysis area a little more clearly. You can also access and activate images from this top bar and press the X on there as well to close images. Down here at the bottom, you'll see an X, Y, and intensity. As you scroll around your image, you can see the X, Y position of your pointer as well as the intensity of your image in that specific spot. Also at the bottom, you'll find the Pan button. When selected, you can move your image and pan around your image when it's zoomed. You can zoom in and out of your image using the magnifying glass buttons at the bottom next to Zoom. From the Contrast module, you can adjust the contrast of your image, including the black and white levels of your image, as well as the gamma. You can choose to autocorrect the contrast of your image, invert the colors of your image, or detect saturation within your image. When you have a multiplex image, you'll also be able to select the channels that are viewed and or adjusted. When you have a multi-channel image open, the option for a channel viewer also appears. Channel viewer opens up a little viewer window that allows you to view each channel separated to see if you have any bands hiding. When you open an image, you will automatically be brought to the Image Adjustment menu. From ROI, you can select areas of your image, adjust the green box to select the area that you desire, and you can copy, paste, or crop that selected area. From Image Orientation, you can rotate your image 90 degrees left or right. You can arbitrarily rotate or flip it horizontal or vertical. If you created a multiplex image and need to align the layers of your image, you can use pixel alignment by selecting the channel that you'd like to align and nudging it using the arrows. Resize allows you to resize your image according to your desired parameters and save the resized image as a publication file. Annotations allows you to add annotations to your image file. You can add shapes, lines, drawings, text, and numbers to your file. You can adjust the color and the thickness of your shape or text, change the font of your text and the size of your font. You can also copy, paste, undo, redo, delete, rotate, and change the alignment of any annotations you add. When you're finished annotating your image, you can hide the annotation so that it's not in the way during analysis. You can also save your annotations as an overlay so that you can load it onto subsequent images. The info button here allows you to see any image info for the image that you have open. Within this basic version of AzureSpot Q, you have access to the intensity analysis tools. With intensity analysis, you can draw shapes over regions of interest by clicking the desired shape and clicking and dragging over your image. If you'd like to create a series of multiple bands in a row that are all the same size, you can use the multi-ROI draw tool by entering the number of bands that you would like, clicking the image and dragging across to create a row of bands. Or if you have a shape that you have drawn and like that shape, you can copy it and use the multi-region paste to paste it to different areas of your image. Once shapes are drawn, if you adjust the size of a shape and would like to apply that change to all shapes within your image, you can apply that shape here. You can also single copy and paste instead of multi copy and paste, and delete. Clicking on the info buttons within intensity analysis will bring up a little more information on the intensity analysis tool. Down here at the bottom, you have the option to invert measurements. This can be useful if you have opaque bands in your image and need to invert the intensities. After intensity analysis, we have background subtraction. There are several background subtraction methods offered within AzureSpot Q, including local, control area, surface minimum, constant, and none, which applies no background subtraction to your image. You can click on any of the background subtraction methods to view a description of that background subtraction method. Quantity calibration allows you to use any known standards within your image to calculate the quantity of your unknown samples. So if you have run a serial dilution of a known protein, you can add those values here and calculate the amount of protein in your unknowns. Molecular weight allows you to define the marker bands within your image, enter the molecular weights of those bands, and use that to calculate the positions and molecular weights of all your unknown bands. Once your analysis is complete, you can save the analysis as an analysis protocol. And once you have saved protocols, you can always open saved analysis protocols to apply them to subsequent images. And that is the overview of all the features and functions of AzureSpot Q.